Welcome to this second video in the series of 3 videos dealing with membranes and membrane function. In the first video, we identified the fluid mosaic model of membrane structure, so you should have a pretty good idea of what this diagram represents. If you didn't get a chance to see that video yet, at the end of this one I'll have a link to go back to that one so you can take a peek at it. In this video, however, I want to go past that point of just the structure of membranes and start dealing with the functions that are involved in satisfying the needs of the cell, and specifically I want to consider diffusion and osmosis. In the third video, we'll get into the specific roles of the proteins and the specific role of the membrane itself in transporting materials across it. But before we get into even diffusion as a topic, I think it's pretty important that we establish that membranes are what we call SPM. That stands for selectively permeable. So we're talking about selectively permeable membranes. Historically, the term actually meant semi-permeable, and that term has been dropped in favor of selectively permeable because this term implies that the membrane plays an active role in determining what can cross it and what can't cross it. If you think of semi-permeable, there's no suggestion in that term that the membrane plays any role at all. It just happens to stop some things and not stop other things. So we're dealing with selectively permeable membranes here. What I said before, and I'll say it again here, is that the cells have constant needs for certain things. For example, oxygen is constantly needed in cells because it's used in cellular metabolism. And carbon dioxide is constantly being produced and it has to be gotten rid of. Same as nitrogenous waste. We think about NH3 ammonia, which cells make and have to get rid of. These little molecules are small enough that they can freely cross the membrane. There's no barrier to them. They can sneak through the molecular structure. And the reason they would even sneak through the molecular structure is because if oxygen is being used up in here, then there's always a deficit of oxygen inside a cell. And whatever the environmental concentration of oxygen is, it's going to be greater than in here. So it crosses the membrane. Same as carbon dioxide. If carbon dioxide is being produced in the cell, it's going to be produced higher than the concentration outside the cell, so it's going to be moving across to the outside. And the same thing is true for ammonia. What I'm describing, without using the term here, is really diffusion. And diffusion happens whether there's a cell or not. So, having said that, let me get rid of the cell. Let's consider diffusion as a topic all by itself. Then we'll bring back the membranes, particularly when we get to osmosis. Diffusion is the movement of particles according to concentration differences. And that's all it is. So if we have a solution, for example, we just talk about any solution, we'd go back to the diagram of a beaker, and we put some water in it, and we dump some stuff in it. Let's say we're going to put some salt in it. The salt's going to wind up in the bottom, and it will dissolve. And as it dissolves, it will slowly, I'll erase the word water here, it will slowly diffuse. There's nothing making it do that. It's just a fact of nature, or we call it the law of diffusion. And then what it says quite simply is that particles will move to satisfy concentration differences. And they'll continue to move until there is no concentration difference. So when this is finally all dissolved, and I'll erase this part, we'll advance it a little bit here. When it's all dissolved and you've left it long enough, the sodium and chlorine ions will be evenly distributed. If they weren't evenly distributed, they would still be moving around a lot in order to make them evenly distributed. And when I say they're evenly distributed, that doesn't mean they're stopped. They're still moving around. If that one moves that way, this one's going to move that way, and that one will move that way, that one will move that way. There is no net difference in concentration. Concentration of sodium chloride ions here is the same as it is over there. That's diffusion. The laws of diffusion, they're going to do that. They randomize themselves as much as they can so that there is no big difference in their concentration. So there's some factors that affect this. Factors that affect the rate of diffusion. And I think I can list about half a dozen of these. Obviously, we're talking about concentrations here. So the more salt you put in, or the more substance you put in, or the more solute you put into a solvent to make a solution, I just hesitate for a second here. A solution is made up of solvents and solutes. The solvent is the component of the solution which is in greater concentration. So there's more water than there is salt. So therefore water is the solvent. The sodium chloride or the salt gets dissolved in it, it's the solute. So back to what I was saying, the first one that we're talking about is concentration. The greater the concentration difference we're talking about, the faster the rate of diffusion. So if we talk about just putting one salt particle in the bottom here, then it's not going to go very fast. But if you put a lot in, it's going to go faster. So the greater the concentration difference, the greater the diffusion. The rate of diffusion, but I'll just write diffusion here. Another one, we're talking about the movement of particles. I'm not going to write down an explanation of them every time. I'll just tell you what they are. It has to do with temperature. As you increase the temperature, it's going to go faster. We're talking about movement. That's kinetic energy. So temperature obviously has an effect. At higher temperatures, particles will go faster. Another one that isn't so obvious when we're talking about water, but maybe if we're talking about something that isn't water. Suppose this was corn syrup, for example. Corn syrup, stuff will dissolve in corn syrup, but not very quickly. It has to do with viscosity. Like how thick is the stuff in the first place? So viscosity, if it's thick, very viscous, diffusion is going to be hampered quite a bit. Number 4 on my list of 6 is size, nature of the particle. By nature I mean shape. Of the solute particle, I mentioned sodium chlorine, and we're just talking about little ions here. Well, if it was sugar molecules, well, sugar molecules are a little bit bigger, they might move a little slower. If we're talking about putting in some big molecules, they're going to randomize themselves, but it's going to take them a lot longer because they're cumbersome, they don't move as quickly through the solution or the solvent at all. Another one that is important here is whether or not there's a charge involved. If we're talking about sodium chlorine and we're talking about ions, so we've got sodium ions and chlorine ions, and if we were talking about a system that had some sort of charge associated with it, so there was something here that may be positively charged. That's not very realistic, but if there's a large say lead ion over here, something like that, that's going to affect it as well, because the chlorine ions will be drawn towards that side, the sodium ions will be repelled away from it because like charges repel, opposite charges attract, that sort of thing. So there could be a charge that's associated with it, so that has something to do with it as well. And the sixth one, the last one on the list, and this one is pretty obvious, especially when I talked about this, if we dump some salt in the bottom and we just let it sit here and eventually it's going to dissolve. Well, right away you might have been thinking, well, yeah, you can do that, why don't you stir it? Because the movement in here, if you're stirring it or you agitate it, you can speed up diffusion. So if you want to put some sugar in your coffee or your tea or something like that, you don't just let it sit in the bottom, you stir it up and it disperses much more quickly, it diffuses throughout. So this is, I'll put stirring, or if you agitate it in cells. You think, well, wait a minute, you can't really stir cells and expect them to do anything, but there is such a thing as cytoplasmic streaming. And cytoplasmic streaming is a natural movement of the cytoplasm in a cell. So the cytoplasm does stream, if you like. So there is a situation where we can talk about that. So that's what makes particles move in the first place, is diffusion. And then, like I say, they move even if you don't have a membrane present. But in this topic, we're really talking about cells, we're really talking about membranes being present. So we're going to change this topic now and take that diagram away and put a different one up here. Let's say we have a cell. Let's say we've got, I don't want to use sodium, sorry. Let's say we've got particles out here, and none of them are inside the cell. We've got water out here, we've got water in here. We have a concentration difference. The laws of diffusion still have to be obeyed. It's a scientific law, it's a principle. Particles will move to satisfy differences in concentration. Particles will move, wait a minute, there's some out here, none in here. So you say, okay, well that's easy, they're going to go in there. The membrane stops them. The membrane's function is to control what can go in or out of cells. So if this is a situation where these particles can go in by diffusion, that's fine, they're going to go in. But if this is a situation where they can't go in, the only way you can satisfy the law of diffusion is if water goes out. Essentially, you dehydrate the cell. This movement of water to satisfy the laws of diffusion is called osmosis. Osmosis satisfies the laws of diffusion. So let me say that we're going to know the law of diffusion here. We've got a bunch of information to record about osmosis. And let me start with just a simple movement of water to satisfy the law of diffusion. Osmosis will occur without a membrane. Maybe there was a bit of a simplification before. Just go back quickly here and consider this beaker, and you've got particles in it, and you've got water in it. The water molecules are going to be moving around in here as well. So they're going to be assisting in satisfying the law of diffusion. But in this case, where we do have a membrane, it's way more pronounced and way more obvious. Now we changed the terminology a little bit here because we don't talk about diffusion so much, although osmosis is the movement. Technically it's the diffusion of water. But we changed the terminology around and we talk about osmotic gradients. So we could have something which is hypertonic. A whole set of terminology comes in here. Hypotonic and hypertonic. These are opposites. The prefix hypo means under. So it's a lesser concentration. This is the abbreviation for concentration. I'll put it in square brackets. When you see square brackets, that means concentration. Hypertonic is a greater concentration. You can remember this because everybody's heard of a hypodermic needle. Well, it goes under the skin, which is the dermis. So hypertonic, lesser or lower, and hypertonic is greater. So taking those two terms, going back to this diagram, this solution out here, remember we're talking solutions, this solution here is hypertonic because it has a greater concentration of these particles in it in comparison to this solution in here. These terms are always used to compare solutions. And so this solution here is hypertonic to this solution here with respect to that substance. This solution here is hypotonic compared to this solution here with respect to those substances. And as water diffuses out, or we say as water moves by osmosis, as water is drawn out or moves out, the concentration of these substances out here will lessen. This becomes less hypertonic. As the water moves out, this solution in here, there's less water in here, so this becomes, well, it was already way hypotonic, but as you take water out, the effective concentration difference between these two things diminishes. Will it ever equal? Will this concentration ever equal this concentration? No, it can't. I didn't put any of these substances in here. Let me put a couple in here. Is it possible that it could ever equal it? Sure, I suppose so, but you'd have to take a lot of water out of here and put it out of here and spread these out a lot more so that the effective concentrations in both regions is the same. Should that happen, we have a thing called isotonic. Iso means same concentration. So that's the introduction to this. What I'd like to do is present some experimental conditions to you to help illustrate this. Then I'm going to bring back the cell and we'll talk about turgor pressure and turgidity and that sort of thing. So just be right back in a second. Welcome back. I've illustrated a commonly used experimental apparatus on the board right now. Let me explain to you what it is. We've got a beaker here, maybe just water. Just a glass tube. Dialysis tubing. What it's possible to do, and what we've done many times, is take dialysis tubing and you tie it off on both ends here, and you tie it around the glass tube. Before you tie it off though, you fill it with some solution. So this might be filled with oh, let's say starch solution. And then you submerge it in the water and you let it sit. Prediction, what's going to happen? Well, logically enough, starch, or if we'd used protein or something like that, doesn't matter. Starch is a very big molecule and it's not going to get out of the dialysis tubing. Dialysis tubing is like a membrane. It'll allow water through, but it won't allow things like starch or protein through. So this can't get out. The result of that is the laws of diffusion have to be obeyed. There's no starch out here. Water is going to go in. Water will dilute that as best it can. And as water continues to dilute it, obviously, the water level is right here in this tube because it's all held in. As water is drawn in, the water will actually rise up in this tube. If the concentration difference was high enough, in theory, you could get the water to squirt out the top of the tube. If you had a tube tall enough, it wouldn't happen, but in theory, that's what would happen. In practice, it doesn't necessarily happen. In practice, it rises up to a certain level. And the reason for that is pretty obvious when you think about the mass of this, because as water climbs up in here, there's a certain mass. The weight of the water, if you like, the weight of the water pushing down is equal to the pressure of the water being encouraged in because of the concentration difference. This, if you could measure this, is equal to osmotic pressure. Like, what is the pressure, or what is the force that's being pushed, or that's pushing the water in here, and you can measure it like that. Osmotic pressure, if you take a look at a different apparatus, I can illustrate it and I'll come back and explain this. Osmotic pressure, this is even a little bit fictitious here. If we take we could take a U-tube. A U-tube is a U-shaped piece of apparatus that has a membrane at the bottom of it. So this could be the membrane here. And I'll put a dotted line and we would label that SPM, selectively permeable membrane, and we can put our water solution in here, or water in there, and then we can add, let's add protein this time. Put protein in there. Let's say we're going to put a 10% protein solution in that side, and we're just going to have water on this side. What's going to happen? Water is going to be drawn through this way. The reason it gets drawn through that way is in order to satisfy the laws of diffusion. To come back to osmotic pressure is exactly the rate and the force of the molecules of water banging on that membrane. Water is going to hit the membrane multiple times, and many of those times it's not going to go through. It's just going to be bounced off. It's not going to hit the protein the right way, or it's going to hit the end of the phospholipid, but water will go through. Osmotic pressure is the rate of collisions of the water molecules on the membrane. Now if we take a look at a different thing, say we had 20% protein solution, the water is going to be encouraged even more because there's a greater concentration difference. They're going to be banging on the membrane even harder, not harder necessarily, but even more frequently. So we're going to have a higher osmotic pressure. If we had a 1% protein solution, that's not going to be very much of a concentration to draw the water over. So we're going to have a lower osmotic pressure because we're not going to get a very big rate of collisions on the membrane. So that's sort of like the experimental apparatus. One thing you can do on your own, and you'll just see the effect of this, you can take a potato, here's a potato, cut a couple of slices of potato, put this one in water, put this one in salt water, and leave them overnight. Then come back in the morning and have a look at them. And you'll see a huge difference in them. And if you think about it, you should be able to predict what's going on here. One of them is going to be firm, and one of them's not going to be firm. That's the next bit of this topic. So let's go back to cells, where we won't use potato cells. We don't need experimental apparatus. Let's go back to any cell. Just take an animal cell, like this. I won't draw the nucleus and all the organelles and that sort of stuff, but here's the cytoplasm in here. The cytoplasm has a certain, what we call turgidity. Or turgor pressure. The cytoplasm does exert a bit of pressure on the surface, the inner surface, pointing out of the cell. That's turgor pressure. Now we can play around with turgor pressure. We can affect it, because if we take this cell and we put it in a salt solution, that's going to affect turgidity. We take a cell and put it in a salt solution, it's going to affect turgidity, because what's going to happen is the water's going to be drawn out. Water's going to leave by osmosis, because this is hypertonic to the cytoplasm. The laws of diffusion are going to draw the water out by osmosis. What happens is the cell loses turgidity. And we say plasmolysis has occurred. The word plasma and lysis means to break. It hasn't really broken, but it's taken the plasma, the fluid part then, the plasma is fluid, and it's leaked out, so we have plasmolysis. If we do the other, if we take this and put it in, I'll use a different cell. If we take a cell here and we put this in distilled water, the effect here is that because there's a myriad of stuff inside here, water is going to be drawn in, because the concentration of anything is greater in here than it is out here. So this is what we call absolutely hypotonic, and we're going to have an increasing turgor pressure. Or cell increases turgidity. This is what we can call deplasmolysis. It's the opposite. One thing we have to not forget here before we leave this topic, we're almost finished with this topic, is that energy hasn't really been involved. There's been no ATP here. All of this is passive, as in doesn't require the input of energy. It's just a law of diffusion. When plasmolysis occurs and when deplasmolysis occurs, one of the things that happens is that the cells don't function the way they should, because they're changed size. Cells are the size they're supposed to be. They function best at the correct size. And if we fiddle around with changing the environment of the cells, we can affect their ability to function. It's the same sort of thing if you put salt on some invertebrates like slugs. It'll cause them to shrivel up. It's the same thing. They will lose their water, and you can actually desiccate, kill a slug that way. I want to leave you with I think two more things. One of them is a contrast between plant cells and animal cells with respect to this topic. And the final one is the effect of cell size and surface area. If we take an animal cell and a plant cell, I'm just going to draw a little chart here. And we put these two cells in a hypertonic solution and a hypotonic solution and look at the differences between the effects. So if we take a hypertonic solution and a hypotonic solution, if we put an animal cell in a hypertonic solution, that means we put the cell in something which causes it to shrivel up, because it's this picture here where we draw water out. The cell shrinks in size. If we put the plant cell in a hypertonic solution, remember plant cells, they've got cell walls, they're typically larger, they've got cell walls. What happens here is, again, they're going to lose water, but the cell doesn't shrink because the cell wall holds the shape of the thing. What happens inside is the protoplasm shrinks. The living part of the cell shrinks. The fluid leaks out, the cytoplasm and the vacuole, which contain a lot of water, that stuff diminishes in size, but the cell wall stays. If we do the opposite, if we take these cells and we put them in hypotonic solutions, this one's going to gain a lot of water and it's going to swell. So it's going to get as plump as it possibly can. It'll become completely spherical if possible, because that's as big as it can get. The plant cell, in contrast, it's going to take in water as much as it can, but you might not see anything. It may look the same. You may not see a difference, because the cell wall doesn't really change shape. It's going to take water in, or allow water to go in, but it's just going to get it, it's going to increase turgidity as much as it can. In human biology, if we were to take this topic and deal with red blood cells, if we put red blood cells in things like distilled water, ethyl alcohol, we will get hemolysis. They break. Heme, that deals with hemoglobin, they actually break. They can burst wide open. If we take the same red blood cells, we're dealing with plasma and circulatory system, and we do this to it, we can often get what we call crenation occurring. That's an N, not cremation. Crenation, and they crenate, which they take on a sort of a notched appearance because of the presence of the hemoglobin in the cell membrane. It holds some of the shape, but it still shrinks overall. The last thing here is dealing with surface area and volume. As a cell swells, its volume increases. Does the surface area increase? No. The surface was already there. But if you swell a cell, or make a cell swell, the volume will increase, surface area will not. The ratio between the surface area and volume ratio will, as a number, whatever the number might be, if we had a calculation here, the number will decrease. Surface area volume ratio decreases as cells swell. The reason it decreases is because that number, that component of the fraction gets larger, this one doesn't change. And The same thing here. If we shrink the cell, the volume decreases. The surface area doesn't change. The surface area is, there's so much of the cell there. The surface area volume as a single number will increase. The ratio of surface area to volume for cell function is a critical thing in determining its function. If it deviates too far from a normal ratio, the cell will cease to function. So there you have it. That topic on diffusion and osmosis was dealing with just how things cross membranes. We're talking about passive movement of things across the membrane. And the next step in this whole topic is the next video, the final video in the series, is getting the membrane involved, the components of the membrane, the actual proteins and what they're doing, and the membrane itself in terms of getting very large substances to cross the membrane. Remember, if you need to go back or you want to go back and have a look at the video on fluid mosaic model, I'll put that link right up here, and my gecko again is always down here. Thank you very much.